Hello, I'm Dean Whiting from Pathology Technology Australia. In my previous COVIDeocast, we talked a little bit about the viral serology tests, the antibody and antigen tests, and how important these are to helping us with detection and management of COVID-19 disease. Today I'd like to talk in a little more detail about the nucleic acid tests, often called the PCR test, to detect the viral RNA. So you would have seen for sure that the samples are collected via a swab, which goes through to the back of the nose or to the throat, or it could potentially also be a sputum sample from lower down in the lungs. It's very important that the swab gets very good connection to the back of the nose or to the throat. And this can be a little bit uncomfortable for many people, but it is really important that the swab makes great contact with the back of the nose to pick up enough viral particles for them to be able to be measured. So that swab goes into a transport container which contains a media, a liquid, effectively, which kills off the virus but preserves the viral RNA. And that goes off to the lab. So in the lab, the first process there is to extract the RNA, extract the nuclear material from the virus. So this extraction process takes place in a number of different ways, but typically there's an entity which recognizes the RNA and binds that to a particle, usually a magnetizable particle, or to a plastic bead or to a plastic surface. And that then helps separate the RNA from all of the other stuff that was in the swab. So with that extracted RNA, we can now subject that to the PCR test, the polymerase chain reaction. So effectively what the PCR test does is through multiple cycles of heat and cool, it replicates up the viral material, sections of the RNA in this case. And once you've been through enough cycles of the temperature increases and decreases, and with the right enzymes and probes and oligos, we're able then to accumulate enough of the very specific material from the virus to then be able to detect it, usually using a fluorescence marker or some other marker, which can then be detected, a signal system which can be detected. So the testing is highly specific for the viral RNA. So the tags or the probes used to bind the RNA have very specific sequences on them which recognize only the COVID RNA. So these are very specific tests. They're also very sensitive because just with a few viral particles handled correctly and through the PCR process, we can multiply them up many times then to be able to do the measurement. The test is not completely foolproof, however. If the probe does not connect well with the back of the nose or throat, then maybe not enough viral particles are collected. If an individual is too early or too late in the infection phase, there may not be much viral material there to be able to pick up in the first place. In some cases, the virus moves further down into the lungs and can be hard to detect. It also depends a little bit on the PCR technology itself and which genes are recognized by the probes in the PCR reaction. And you may sometimes hear of single gene, double gene, 3 genes, 4 genes being detected in the PCR reaction. And it's important to note the mix of these genes because that then gives you an indication of how likely the assay is to pick up the presence of the virus. So a single gene probe is great, but it may miss some versions of the especially early phase detection and late phase detection of the presence of the virus. 2 or 3 genes in the mix means that you're targeting multiple sites on the viral RNA and gives you a better chance of picking it up. But as I say, still not foolproof. There is a low but measurable false negative rate. It's not likely, there is some false positive rate, but it's not a great rate. So there are also a couple of other nuances here too. One is that there are many proprietary PCR tests available now. Most of the large manufacturers manufacture a PCR test for COVID-19 on their proprietary platforms. So these are usually high volume automated systems for measuring quite large numbers of samples. And these proprietary systems have an obligatory trail of consumables, usually plastics, tubes, tips, and sometimes the test kits themselves. So they're configured to be only usable really on the proprietary platform. And there are some really good reasons why you would want to do this, because then a single company, the manufacturer, takes responsibility for the quality of the entire process. So for the extraction, for example, and the detection. So for the detection alone, if you're using a proprietary system, the manufacturer has validated the reagents and consumables for that system and knows that it works extremely well. So there's a big advantage in going with proprietary. But likewise, some of the early work was done with what's called open platform systems, where the probes and primers, so the little bits of DNA or RNA that find and bind to the viral RNA, can be done in open tubes or in plate form, 96-well plate form, on open platforms. This often relies then on the lab itself doing the validation, and it can suffer some vagaries of consistencies of the probes and primers, of the consistencies in how the test is done. But the labs are usually pretty good at this. They have very well thought through processes, very well validated processes. And so really the only drawback of methods like this is that they can be time consuming, and they're not really open to large volumes and large throughputs. So that's the PCR-based testing, the nucleic acid testing for COVID-19 detection. I hope that's been helpful. And until next time, I'm Dean Whiting, Pathology Technology Australia.